Hello everyone, my name is Hendrick. I would like to give a flash talk here at Virtual Great Lake Bioinformatics Conference 2021 of our work entitled Transcriptomics and Proteomics Analysis to Identify Signature Pathways in Colorectal Cancer Progression. To begin, I will start with the fact that colorectal cancer is one of the most common cancer happen in the human worldwide. It also become one of the deadliest cancer occur in the human. For this reason, studying multi-omics data, for example, transcriptomics and proteomics, could be essential to discover signature pathways in colorectal cancer progression in order to give a better understanding on how this cancer can happen from molecular level. Here, we conduct an integrated multi-omics analysis, transcriptomics and proteomics, with the hope to identify differences of signature pathways in colorectal cancer progression from transcript and protein domains. To accomplish our previous objective, we used two independent colorectal cancer studies that contain tumor and adjacent normal samples as our datasets. The first study for transcriptomics contained 10 pairs tumor tissues with their adjacent normal. Meanwhile, the second study represents proteomics of 8 pairs tumor tissues with their adjacent normal. This diagram shows the outline on how we conduct our integrated analysis. We start by selecting data of transcriptomics from Gene Expression Omnibus and proteomics from Proteome Exchange public data repository with its data accession number stated in the bracket. Then we process the data. For transcriptomics, we use GO2R to perform statistical tests on identifying differentially expressed transcripts between two groups, tumor and normal. As a result, we had a total of more than 2,000 differentially expressed transcript IDs. Meanwhile, for proteomics, we use MaxQuant to quantify its label-free quantification intensity. Then, Parse software was used to select significant proteins. As a result, we had more than 1,000 protein IDs with their normalized intensity value. Next step, for gene set arrangement analysis, we use KEGG pathway information to identify signature pathways in colorectal cancer. All transcript and protein IDs were collapsed to gene symbol to make the results comparable each other. Then we use cytoscape to visualize the results as networks and find the overlap KEGG terms in these two domains. Here we show the gene set arrangement result of its specific phenotypes. The Venn diagram on the bottom right shows the correlation between the identified KEGG terms. In transcriptomics, we found 23 enriched KEGG terms, 10 for tumor, as shown on purple circle, and 13 for normal, as shown on yellow circle on the Venn diagram. Meanwhile, we found another 27 enriched KEGG terms in proteomics, 16 for tumor as shown in green circle, and the rest of 11 for normal as shown in red circle on the Venn diagram. Here we show the visualization of the KEGG terms with the cytoscape tool. On the top, there are 23 nodes that represent enriched KEGG terms available from the transcriptomics data. Meanwhile, on the bottom, there are 27 nodes that represent enriched KEGG terms available from the proteomics data. Note with the red indicates terms that enrich more in tumor samples, while note with blue indicates terms that enrich more in normal samples. Darker node color indicate higher position of KEGG terms relative to that node in our list based on the normalized enrichment score. Next, we edit the available networks by removing all KEGG terms which are not appear in both two domains and just keep the terms that appear in both two domains along with their age information. The results, as you can see here, some age information are consistent in both domains, as marked with orange circle to create specific subnetworks of neurodegenerative disease, since it involves Alzheimer's, Huntington, and Parkinson's disease together. Meanwhile, some age information are just persist in protein domain, but not in transcript domain, as shown on green circle on another subnetwork. To summarize my talk, in our work of integrated transcriptomics and proteomics study, we found several signature pathways that underlying in colorectal cancer progression. The first is focal adhesion pathway that related to pathway in cancer, since it preserved in the protein level rather than transcript level. The second is oxidative phosphorylation pathway that relate to other neurodegenerative disease, since it preserved in both transcript and protein level. These two pathways may become a great potential target to treat colorectal cancer. Thank you very much for listening.